The topic of today's lecture is genetic, hormonal, and environmental controls of growth and development. The ultimate control of plant growth and development is genetic. If the genes are required for development of a particular trait, for example, the shape of the leaf, the color of the flower, or the type of root system are not present, that characteristics does not develop. When a particular gene is present, its expression, that is, how it exhibits itself as an, as an observable feature is an organism, is influenced by several factors including signals from other genes and from other environment. The location of a cell in the young plant body also has a profound effect on gene expression during development. Chemical signals from adjacent cells help a cell perceive its location within the plant body. Each cell's spatial environment helps determine what that cell ultimately becomes. Growth and development, including plants' responses to various changes in the environment, are regulated by plant hormones, organic compounds that act as chemical signs between cells. Environmental cues such as changing day length and changing temperatures exert an important influence on gene expression and hormone production, as they do on all aspects of plant growth and development. The initiation of sexual reproduction is often under environmental control, particularly in temperate latitudes, and plants switch from vegetative growth to reproductive growth after receiving appropriate signals from the environment. Many flowering plants are sensitive to changes in the relative amount of daylight and darkness that accompany the changing seasons, and these plants flower in response to those changes. Other plants have temperature requirements that induce sexual reproduction. Plants then continually perceive information from environment and use this environment, environmental information to help regulate normal growth and development. Plants exhibit movements in response to environmental stimuli such as light, gravity, and touch. A plant may respond to such an external stimulus by directional growth, that is, the direction of growth depends on the direction of the stimulus. Such a directional growth response, called a tropism, results in a change in the position of a plant pot. Tropisms are irreversible and are positive or negative, depending on whether the plant grows toward the stimulus, this is a positive tropism, or away from it, a negative tropism. Tropisms are under hormonal control, which is discussed later. Most growing shoot tips exhibit positive phototropism by bending toward light. This growth response increases the likelihood that stems and leaves receive adequate light for photosynthesis. Blue light, that is, with a wavelength between 400 and 500 nanometers, triggers the bending response of phototropism. For a plant to have a biological response to light, it must contain a light-sensitive substance called a photoreceptor to absorb the light. The photoreceptor that absorbs the blue light and triggers the phototropic response is and also other responses uh, is a family of yellow pigments called phototropins. Most stem tips exhibit negative gravitropism by growing away from the center of Earth, whereas most root tips exhibit positive gravitropism. The root cap is the site of gravity perception in roots. When the root cap is removed, the root continues to grow, but it loses any ability to perceive gravity. Special cells in the root cap have starch-containing amyloplasts that collect toward the bottoms of the cells in response to gravity. And several lines of evidence indicate that these amyloplasts initiate the gravitropic response. If the root is reoriented, as when a potted plant is laid on its side, the amyloplasts tumble to a new position, always settling in the direction of gravity. The gravitropic response bending occurs shortly thereafter and involves the hormone auxin. Despite the movement of amyloplasts in response to gravity, their role in gravitropism remains an open question. Mutant Arabidopsis plants with few or no amyloplasts in their root cap cells exhibit some gravitropic response when placed on their sides, suggesting that roots do not necessarily need amyloplasts to perceive gravity. Thigmotropism is growth in response to touching an object. An example of thigmotropism is the twining or curling growth of tendrils or stems which help attach a climbing plant, such as a vine, to some type of support. Like phototropism and gravitropism, thigmotropism involves auxin. A plant hormone is an organic chemical compound that acts as a chemical signal, eliciting a variety of responses that regulate growth and development. The study of plant hormones and their effects is challenging because hormones are effective in extremely small amounts. In addition, the effects of different plant hormones overlap, and it is difficult to determine which hormone, if any, is the primary cause of a particular response. Plant hormones may also stimulate a response at one concentration and inhibit that same response at a different concentration. For many years, biologists studied five major classes of plant hormones: auxin, gibberellin, cytokine, ethyl N, and abscisic acid. More recently, researchers have uncovered compelling evidence for a variety of other signaling molecules such as brassinosteroids, oligosaccharides, jasmonates, salicylic acid, and cysteamine. Charles Darwin, uh, the British naturalist best known for developing the theory of natural selection and explain evolution, also provided the first evidence of the existence of plant hormones. The experiments that Darwin and his son Francis performed in the 70s involved phototropism in 70s of the 19th century uh, in newly germinated canary grass seedlings. As in all grasses, the first part of a canary grass seedling to emerge from the soil is the uh, coleoptile, a protective sheath that encircles the stem. When coleoptiles are exposed to light from only one direction, they bend toward the light. The bending occurs close to the tip of the coleoptile. The Darwins tried to influence this bending in several ways. For example, they covered the tip of the coleoptile as soon as it emerged from the soil. When they covered that part of the coleoptile about where the bend would normally occur, the plants did not bend. On other plants, they removed the coleoptile tip and found that bending did not occur. When the bottom of the coleoptile, where the curvature would occur, was shielded from the light, the coleoptile bent toward light. From these experiments, the Darwins concluded that some influence is transmitted from the upper to the lower part, causing it to bend. In 1920s, Fritz Wendt, a young Dutch plant physiologist, isolated this influence from old coleoptiles. To do so, he removed the coleoptile tips and placed them on tiny blocks of agar for a period of time. When he put one of these agar blocks squarely on a decapitated coleoptile, normal growth resumed. When he placed one of these agar blocks to one side of the tip of a decapitated coleoptile in the dark, bending occurred. This bending indicated that the substance had diffused from the coleoptile tip into the agar, and later from the agar into the decapitated coleoptile. Wendt named this substance auxin, from the Greek word that means enlarge or increase. Several naturally occurring auxins have been identified, and some artificial auxins have been synthesized. The most common naturally occurring auxin is indolacetic acid, or IAA. The movement of auxin in the plant is polar or unidirectional. Auxin moves downward along the shoot root axis from its site of production, usually the shoot apical meristem. Young leaves and seeds are also sites of auxin production. Auxin's most characteristic action is promotion of cell elongation in terms and uh, coleoptiles. This effect, apparently excited by acidifying the cell walls, increases their plasticity, enabling them to expand under the force of the cell's integral turgor pressure. Turgor pressure is the pressure that develops against a cell wall when water moves into the cell by osmosis. Auxin's effect on cell elongation also provides an explanation for phototropism. When a plant is exposed to a light from only one direction, the auxin moves laterally to the shaded side of the stem before moving down the stem by polar transport. Because of the greater auxin concentration on the uh, shaded side of the stem, the cells there elongate more than on the cells on the lightest side, and the stem bends toward the light. As mentioned earlier, auxin is also involved in gravitropism and thigmotropism. Auxin exerts other effects on plants. For example, some plants branch out very little when they grow. Growth in these plants occurs almost exclusively from the shoot apical meristem rather than from auxiliary buds, which do not develop as long as the terminal bud is present. Such plants are said to exhibit apical dominance, the inhibition of axillary bud growth by the shoot apical meristem. In plants with strong apical dominance, auxin produced in the apical meristem exhibit axillary buds near the apical meristem from developing into actively growing shoots. When the apical meristem is pinched off, the auxin resource is removed and axillary buds grow to form branches. Apical dominance is often quickly re-established, however, as one branch begins to inhibit the growth of others. Other hormones, ethylene and cytokinin, both discussed uh, later, are also involved in apical dominance. As with many physiological responses, the interactions of these hormones rather than the action of one hormone alone may be responsible for apical dominance. Auxin produced by developing seeds stimulates the development of the fruit. When auxin is applied to certain flowers in which fertilization has not occurred and therefore in which seeds are not developing, the ovary enlarges and develops into a seedless fruit. Seedless tomatoes are produced in this manner. Auxin is not the only hormone involved in fruit development, however. Some manufactured or synthetic auxin have structures similar to that of IAA. So the IAA is the indole acetic acid. A synthetic auxin is used to stimulate root development on stem cuttings for asexual propagation, particularly of woody plants with horticulated importance. The synthetic auxins 2,4-D and 2,4,5-D are used as selective herbicides. Uh, these compounds kill plants with broad leaves but, for reasons not completely understood, do not kill grasses. Although both herbicides are similar in structure to indoleacetic acid, they disrupt the plant's normal growth process. Because many of the world's most important crops are grasses, for example, wheat, corn, and rice, both 2,4-D and 2,4,5-D can be used to kill broadleaf weeds that compete with these crops. The use of 2,4,5-D is no longer allowed in some countries because of its association with dioxins, a group of mildly to very toxic compounds formed as byproducts during the manufacture of 2,4,5-D. One of the many controversial aspects of the Vietnam War was the defoliation program carried out by the United States in South Vietnam. From 1961 to 1971, the U.S. sprayed Agent Orange, a mixture of the herbicides 2,4-D and 2,4,5-D over large areas of South Vietnam to expose suspected hiding areas and destroy crops planted by the Viet Cong and North Vietnamese troops. The negative impact of these herbicides on the development of still being felt today. Many ecologically important mangrove forests and commercially valuable hardwood forests were destroyed. In addition, the herbicide sprays caused health problems in the native people and members of the U.S. military who were exposed to them in the Vietnamese jungles. The herbicides contained traces of dioxin, which is now known to cause birth defects in animals. Reportedly, the number of birth defects and stillbirths in Vietnam increased during the herbicide spraying. Also, American veterans who were exposed to high levels of herbicides have more health problems than do other veterans, including certain types of cancer and skin disease. In the 1920s, a Japanese biologist was studying a disease of rice in which the young rice seedlings grow extremely tall and spindly, fall over and die. The cause of this disease, dubbed the foolish seedling disease, was a fungus that produces the chemical substance gibberellin. Not until after World War II did scientists in Europe and North America learn of this work, and the hormone was found to be one of a class of compounds collectively called gibberellins. During the 1950s and 1960s, studies in the United States and Great Britain showed that healthy plants also produce gibberellins. Gibberellins are hormones involved in many normal plant functions. The symptoms of the foolish seedling disease are caused by an abnormally high gibberellin concentration in plant tissue. Because both the plant and the fungus are producing gibberellin. Currently, dozens of naturally occurring gibberellins are known, although many are probably inactive forms. There are no synthetic gibberellins. Gibberellins promote stem elongation in many plants. When gibberellins are applied to a plant, particularly certain dwarf varieties, the elongation may be spectacular. Some corn and pear plants that are dwarfs as a result of one or more mutations changes in the genetic material grow to a normal height when treated with gibberellins. Short stems, high-yielding varieties of wheat uh, have short stems because they have a reduced response to gibberellins. Gibberellins are also involved in bolting, the rapid elongation of floral stalk when that occurs naturally in many plants when they initiate flowering. Gibberellins cause stem elongation by stimulating cells to divide as well as elongate. The actual mechanism of cell elongation differs from that caused by auxin, however, and recall that indole acetic acid induced cell elongation involves acidifying the cell wall. The gibberellin-induced cell wall acidification does not occur. Instead, gibberellin increases the cell wall's ability to expand. Gibberellins affect several reproductive processes in plants. They stimulate flowering, particularly in long-day plants. In addition, gibberellins can substitute for the low temperature that biennials require before they begin flowering. If gibberellins are applied to biennials during their first year of growth, flowering occurs without exposure to a period of low temperature. Gibberellins, like auxin, affect fruit development. As mentioned in the previous lectures, agriculturalists apply gibberellins to several varieties of grapes to produce larger fruits. Gibberellins are also involved in the seed germination of certain plants, cereals, and other grasses. In a classic experiment involving barley seed germination, researchers showed that the release of gibberellin from the embryo triggers the synthesis of an enzyme that digests starch in the endosperm. As a result, glucose becomes available for absorption by the embryo as it resumes growth. In addition to mobilizing food reserves inside a newly germinated seed, application of gibberellin substitutes for low temperature or light requirements for germination in such seeds as lettuce, oats, and tobacco. During 1940s and 1950s, researchers were trying to find substances that might induce plant cells to divide in tissue culture, a technique in which cells are isolated from plants and grow in a nutrient medium. They discovered that cells divided when placed in the culture containing coconut milk. Because coconut milk has a complex chemical composition, investigators did not chemically identify the division-inducing substance for some time. Finally, researchers isolated the active substance from a different source. They called it cytokine because it induces cell division, or cytokinesis. In 1963, researchers identified the first plant cytokinin in corn, and since then similar molecules have been identified in other plants. Biologists have also synthesized several cytokinins. Cytokinins promote cell division and differentiation of young, regularly specialized cells into mature, more specialized cells in intact plants. They are required ingredient in many plants tissue culture media because they must be present in all dividing plant cells. In tissue culture, cytokinins interact with auxin during the formation of plant organs such as roots and stems. For example, the tobacco tissue culture, a high ratio of cytokinin to auxin induces shoots to form, whereas a low ratio of cytokinin to auxin induces roots to form. Cytokinins and auxin also interact in the control of apical dominance, in which the shoot apical meristem suppresses the growth of axillary buds. Here their relationship is antagonistic. Auxin inhibits the growth of axillary buds and cytokinin promotes their growth. One effect of cytokinins on plant cells is to delay the aging process. Plant cells, like all living cells, go through a natural aging process known as senescence. Senescence is accelerated in cells of plant pods that are cut, such as flower stems. But botanists think plants have to have a continual supply of cytokinins from the roots. Cut stems of course lose their source of cytokinins and therefore age rapidly. Despite their involvement in many aspects of plant growth and development, cytokinins have not been approved for many commercial applications other than plant tissue culture. However, in the mid-19th, molecular biologists used genetic engineering to develop tobacco plants that produce more cytokinin and therefore live longer. This discovery has the potential to increase the longevity and productivity of certain crops. During the early 20th century, scientists observed that the gas ethylene has several effects on plant growth, but not until 1934 did they demonstrate that plants produce ethylene. This natural hormone influences many diverse plant processes. Ethylene inhibits cell elongation, promotes seed germination, promotes apical dominance, and is involved in plant responses to wounding and invasion by disease-causing microorganisms. Ethylene also has a major role in many aspects of development, including fruit ripening. As a fruit ripens, it produces ethylene, which triggers an acceleration of the ripening process. This induces the fruit to produce more ethylene, which further accelerates ripening. The expression of one rotten apple suppose the lot is true. A rotten apple is one that is overripe and produces large amounts of ethylene, which diffuses and triggers the ripening process in nearby apples. Humans use ethylene commercially to uniformly ripen bananas and tomatoes. These fruits are picked while green and shipped to their destination, where they are exposed to ethylene before they are delivered to grocery stores. Ethylene, along with auxin, is involved in leaf senescence and abscission. As the leaf ages, uh, the level of auxin in the leaf decreases. Concurrently, cells in the abscission layer at the base of the petiole begin producing ethylene, which in turn stimulates weakening of the cell walls in this location. So that is all for today. Thank you for attending, and see you 2 weeks after, or at the lecture, and then also at the practice. And any questions so far?
Mhm. No, thank you.
No? Okay, then see you today or the next week. Bye bye.